how to take pictures and videos of fast-moving microorganisms. Hi, hello and welcome, Microbehunter here, and today I received a question, and it's about taking pictures and making videos of bacteria that move very quickly. This can be a little bit difficult sometimes, and in this episode I want to talk about this. I want to give you some advice on what you can do to increase the chance of actually getting a good video and or pictures. But I'm a little bit afraid it's going to be a bit technical, this podcast episode, so you got to bear with me. But I hope in any case that it's going to be good advice for you if you have problems. So what I'm going to do now is I'm going to read the question first, then I'm going to explain to you what the problems could be, and then I have a whole list of different things what you could try to tweak. Okay, so here is the question. We're trying to photograph speedy spirilla, both stills and video, but with no success. By the time the video is activated, they're in another universe. We have a USB 3.0 high-definition camera. Any suggestions to gain success? Thank you. Well, thank you for the question, and yes, I do have a whole range of suggestions here. But first I would like to explain you what the possible problems are, and there are three things that you might struggle with. The first thing is that those spirilla, these are spiral-shaped bacteria, they rotate like a corkscrew and they move relatively fast horizontally. And when they move fast horizontally, they might move out of the field of view very quickly. So by the time that you activated the camera, essentially they're already gone, just like you mentioned in the question. Okay, so it's the horizontal movement that's a problem. The second problem is the vertical movement. Especially at a high magnification, the depth of field is low, and this means that those spirilla, when they move vertically, they move immediately out of focus. And then you cannot see them anymore. And other bacteria might move into focus. They were not there before, and then you can see them, and then they're gone again. So it's very chaotic. And the last problem is one that's called motion blur, and that has to do with the camera settings, especially if the shutter speed is too slow. That means you're not able to freeze them, but rather the problem is that they kind of look a little bit fuzzy because they're moving along. Okay. And you want to actually freeze the image. So these are some possible problems that we have to deal with, and it's possible to deal with them, and that's why basically I'm going to go through the points right now. Now, if you want to take still images of the spirilla, and if still images are all that you need, then I highly recommend that you do so-called a heat-fixed slide. And what you do is the following. You take the water sample with the spirilla, you put it on a microscope slide, and you allow the water to evaporate at room temperature. And what's going to happen then is that the spirilla, the bacteria, are then on the microscope slide. And then you pull the microscope slide 2 or 3 times quickly through a gas Bunsen burner flame. Okay, what this is called heat fixing. Of course, with the bacteria side facing away from the flame, how hot should the slide be? You should still be able to hold it in your hand. Okay, so you should not burn yourself, but it should be pretty hot. Okay, but if you're just barely able to hold it in your hand, then that's fine. And this will do the following. It will bake the bacteria to the glass slide, a little bit like an egg sticking to a frying pan, because the proteins, they will cross-link with the glass slide. And then what you do is you directly put immersion oil on the bacteria on the slide without, and that's important, without adding a cover glass. And then you use oil immersion. Okay, if this does not work for you because the bacteria are too transparent, you can also add a staining step. Usually methylene blue will work, but this actually will allow you to take pictures of the spirilla which are not moving. Okay, because you just want to have still images, right? And this is actually the first thing that I would recommend if still images are that what you want to have. Otherwise, I would say we have to have a look at how to capture videos, okay? And that's a whole different issue. My suggestion is that even if you want to have pictures, still images, go for video. And the reason is because afterwards you can always extract the individual frames in the individual pictures. Because when you take a video, when you make a video, then you're capturing one picture, 25 pictures or 30 pictures per second. And then you have a lot of pictures to choose from. You can extract the individual frames from a video either over a video editing program, or what you do is you just display the video, you pause the video and you make a screenshot, and then you also have a picture. So there are different possibilities, but that is actually for moving microorganisms. I would actually only capture a video and then worry about getting the pictures out later. Okay, because by the time you've pressed the shutter button on the computer, it's already gone. Yeah? A possible disadvantage could be that the resolution of the video is lower than with still images, but that doesn't matter. Really, because it's still high enough, and at that high magnification, it's already generally the microscope's resolution that is limiting. So any higher camera resolution really is not going to give you more image information anyway. So I would say just go for video and worry about the pictures later. But I'm going to now do the following. I'm going to step you now through a couple of points here. The first thing that you want to do is you want to limit the vertical movement of the bacteria so that you don't have this focusing problem. And you can do that by already preparing the specimen, usually the water sample which contains the bacteria. Make sure that you use very, very little water. Okay, a tiny amount of water. And this basically means that there is going to be a very thin film of water between the cover glass and the microscope slide. And this kind of almost sandwiches the bacteria between cover glass and microscope slide. Even if you use very little water, bacteria are so small that they still might move vertically a little bit because the cover glass is also not completely flat. Okay, so that is one thing. And the second thing is when you prepare the slide, make sure that there is no dust or dirt or some debris or plant material or whatever in this water sample on the slide, because they too are too large and they will lift up the cover glass a little bit. And this means the cover glass won't stick all the way down, and this basically again increases the space that the bacteria have to move vertically. So really make sure that you, if possible, only have the water sample with the bacteria, and try to remove all solid material that you see, maybe using fine tweezers or I don't know, or use maybe your fingernail and press against the cover glass to make sure that it's really flat. Okay, so that's basically one suggestion that I have. Not normally, but when you take pictures of bacteria, what you usually do is you don't even use a cover glass. You simply allow it to dry, and then you stain it. But that's not what you want to do, because you want to actually capture the movement, right? I assume. Yeah. Otherwise, if this is not the point, if you do not want to capture the movement of them, then I would say that you just dry the slide, and then they're basically flat on the microscope slide anyway. So that is one thing that you can do. The next way that you can increase the depth of field is by completely closing the condenser of your microscope. And closing the condenser will have a couple of interesting effects. Yes, it increases the depth of field. So even if they move a little bit vertically, they might still be in focus, okay? The second thing is the contrast goes up because you are introducing certain diffraction patterns. And this means that the spirilla or the bacteria, in this case, they can be seen better. However, there is a side effect, and this means that the light intensity will go down. And this has again an effect on the required exposure time. I'm going to talk about this later. But close the condenser simply to get the highest depth of field. Okay, so the next suggestion that I have is that in many cases people will use a high magnification objective, like for example 100 times oil immersion objective, to take pictures of bacteria. But I would not do that. The reason is that the higher you go with the magnification, the lower the depth of field, again, but also the smaller the field of view. So this means that essentially they're going to be moved horizontally out of the field of view very quickly, and it's going to be difficult for you to chase after them because you don't know where they are now. So my suggestion is that you use a lower magnification, like many microscopes have a 40 times objective, or maybe a 60 times objective. Maybe not so many microscopes have that, but that's also a possibility. And when you use those objectives, you get a larger field of view, also a larger depth of field, and also a little bit more image brightness. And this actually might be better than using a higher magnification, which actually gives you a higher resolution, but you have other problems, okay? So you might go for a lower magnification first, okay? So another suggestion that I have a little bit, which just came into my mind, is kind of to avoid the problem of bacteria moving out of the field of view is to use a higher concentration of bacteria, because then you always have some of them at least in the field of view, okay? So after we've done this, we can now deal with the large topic of camera settings, okay? Because there is also something that you can do there to increase the success. You've got a USB 3.0 high definition camera. That's nice to have. A USB 3.0 allows you for fast data transmission between the camera and the computer, provided that you actually have plugged it in also into a USB 3.0 port. That's really important, because if you plug it in into a slow port on your computer, the camera will switch down to USB 2.0. So just please be careful about that. But concerning the camera settings, you have to understand the following, that a high-speed data transmission between camera and computer only has a, how shall I put this, is nice to have, is good to have, but might not always solve the problem, because the problem is you need a fast shutter speed. Okay, so, and that is the thing, and that's what we're going to be dealing here right now, okay? So first of all, make sure that you adjust the fastest frame rate and also the fastest shutter speed. These are two different things, okay? I mean, they're indirectly connected a little bit, because you cannot have a fast frame rate if the shutter speed is too slow, but try to, let's say, use a frame rate of, let's say, 24 to 25 or 30 frames per second. That is the standard things here. And then you go for a very short shutter speed. And what's going to happen then is the following, that you're going to see that also the image brightness goes down. If the image brightness is too low, then you, of course, turn up the light intensity of the microscope. If this doesn't work because you're already at the maximum intensity, then you have to go up with the so-called the gain of the camera. Basically what you're doing is you're increasing the camera sensitivity, and there should be a setting somewhere in the software where you can adjust the gain. So you just put the gain level up, and this actually makes the image brighter again, with the unfortunate side effect that also the noise, the image noise, will also increase. The image grain will also increase. Okay, so this is, but still you want to go with a fast frame rate. Let's say, I don't know, maybe it's able to do 30 frames per second, and also a fast shutter speed. If you're not able to go up with the frame rate, then I suggest that you go down with the image resolution, with the number of pixels. Because if you have a lower number of pixels, if basically the video is smaller in size, then the USB port is also able to transmit more pictures, and then you can also have a faster frame rate. So go down with the overall image resolution, to go up with the frame rate. Maximum light in the microscope. If too dark, still increase the gain. If still this isn't a solution, then try to simply keep the image dark as long as you're able to see still something. You're basically underexposing or deliberately underexposing it, and then later on in an image editing program or video editing program, you are going to later on increase the brightness of the image. Okay, so what we're trying to do right now is we're really trying to make sure that we capture as much image information as possible, even if we don't see it, because we're going to try to get it out later on in a post editing process. Okay, so in worst case you just underexpose it and you see what you can do afterwards with it. Okay, so these are all of the camera settings, and there is still another possibility now, last but not least, to actually physically slow down the bacteria. And I have 3 suggestions here what you could do. I have to admit I did not try all of them, okay? But so maybe you can report back maybe if you're successful here. What you have to do is you have to add a substance which is viscous, which is very viscous, so that the bacteria, which are corkscrewing, because the spirilla, they're turning like a corkscrew, and this slows them down. You might add a little bit of a substance called glycerol. These are readily available. It's readily available, and it's very viscous. It can be mixed with water. What I read, however, is that it might have the disadvantage that because it will actually it's osmotically active. So this means it's going to pull out the water of the cells. So you have to see to what extent the spirilla actually react negatively to the glycerol because of water loss. So you got to simply experiment. Yeah. So glycerol is one thing that has been used but has the disadvantage of actually pulling out water. And this might harm the cells, or maybe not. We'll see. The second thing is a commercial solution, a commercial product. It's called ProtoSlow. ProtoSlow, SLO. It's one word. It's a commercial product, and this is a solution that you add to slow down protozoa. Protozoa are eukaryotic microorganisms, so they are much larger. But maybe ProtoSlow also works for bacteria. Okay, you might try this. And last but not least, also something that I did not try out yet, that you might try is starch. And what you do is you take some potato starch or corn starch and you boil it with a little bit of water. And this also makes some kind of like a sticky glue-like substance. And this is also very viscous. And you might also try to add this. But if you add too much starch, then it's going to be so thick that it won't mix well. So maybe not too much starch, but you have to boil it to make sure that it really becomes a homogeneous solution. And if you mix this now with the water containing the spirilla, maybe this also slows them down. I think the starch solution should not be osmotically active. Okay, it's a polysaccharide after all. Yeah. So these are basically the suggestions that I have here. And because there were so many points here, I'm just going to summarize everything again very briefly. So you see, you have to work on multiple levels here. The first thing is to work on the specimen preparation. Use very little water. Make sure that you have a very thin water film beneath the cover glass. Work on the microscope by closing the condenser. Also, maybe not using the highest magnification. That's the second thing. So to increase the depth of field and to increase the field of view. The next thing would be to tweak the camera settings to make sure that you have a fast frame rate and also a fast shutter speed. The fast shutter speed is to reduce the motion blur, and the fast frame rate is there so that you can capture many pictures one after the other, so that you have more to choose from. And if this doesn't work because the image is underexposed, then either you just leave it underexposed or you turn up the gain. But of course, first you have to turn up the microscope lamp, okay? And if this doesn't work, then you increase the gain in the camera, so you have to go all manual settings here. And last but not least, if you've tried all of these methods here, then I would actually try to physically slow down the spirilla, okay? Either with glycerol, with a commercial substance called ProtoSlow, and or with a starch solution, okay? And I hope that this actually will help you a little bit here, and that this increases also the chance of you being successful. But you might always need a little bit of patience. And I would say that if you're patient, and it's also a little bit of luck, really. It's also in my case, when I make my YouTube videos, I mean, I'm sometimes filming quite a lot, and there's lots of luck involved, and I'm just showing those clips in those parts where you can actually see something nicely. I'm not showing you all of those videos which did not work, or did not work as well, obviously. So you see, you gotta simply collect a lot of videos here, and then if you're lucky, you're going to have some videos that actually show the things that you want to show quite nicely, okay? A very last comment that I want to say, is not related directly to your question, but I do recommend, if you want to take pictures of moving bacteria and so on, generally having phase contrast is a nice thing to have, because it really increases the contrast, and you simply also have it much easier to see the structures. Yeah, but again, not everyone has that, and if not, then it also works by simply completely closing the condenser to increase the contrast this way. Yeah. So I think that's enough for today. I wish you all the best. Happy microbe hunting as always, and see you around next time. Bye bye.